Unit 3.9, we do practice. Serine and glycine are both amino acids where serine is polar and glycine is nonpolar. The two amino acids are not labeled and must be identified. Part A asks us to explain why thin layer chromatography is more appropriate than paper chromatography in this case. And to help us, underneath we have our structures of our serine and our glycine. So remember, the two biggest differences between paper chromatography and thin layer chromatography is that paper chromatography is going to be most useful with colored solutions, whereas thin layer chromatography we can use for solutions that aren't colored, and we can use UV light to show the separation in solutions that aren't colored. What we didn't mention in class is that with paper chromatography, we're most often going to be working with transition metals or iodine. So when we look at our structure for serine and glycine, we can tell that we don't have those transition metals or iodine present, and there is no mention in the question about these solutions being, or these substances being, colored. So we can reasonably assume that these are non-colored substances. So in that case, thin layer chromatography is going to be more appropriate. But they've already told us that, so we need to explain it fully. So a complete explanation would be something like paper chromatography is going to separate colored mixtures, and these amino acids don't seem to be colored. There's no mention of color. Whereas thin layer chromatography can separate and identify colorless solutions using UV light, so that would be the more appropriate method choice in this particular instance. So for part 2 of this they're asking us to describe how to set up a thin layer chromatography experiment that will help identify the two amino acids. So thin layer chromatography is very similar in setup to paper chromatography. So for our thin layer chromatography, what we're going to do is we're going to take a thin sheet of either plastic or glass and coat it with a very polar substance. And the polar substance we've been mentioning in class is silica gel. So we're going to coat that plate, whatever it's made of, with a polar substance. In our case, we can use silica gel as our example. It would be beneficial to have a particular substance in mind as an example. So either alumina or silica gel, but they're both going to be very polar. And then the two samples are placed near the bottom of the sheet and labeled in pencil. So that step would be like our step 1. And for step 2, we're now going to place this sheet with our labeled samples into a container containing a non-polar solvent that will fluoresce in UV light. And so we want to place it into that container so that the bottom of our plate is just touching the solvent. So our solvent's not coming up too high, and our solvent isn't touching our samples at the bottom. So the solvent is touching our plate, but isn't touching our samples quite yet. For step 3, we're going to allow time to pass until the leading edge of the solvent is near the top. We're not going to allow it to go all the way to the top of our plate. And then for step number 4, we're going to shine our UV light onto our sheet and mark in pencil the dark spots that would indicate our separation. And then finally, so that portion is actually that finishes the actual thin layer chromatography experiment. So everything from here would be analysis, but worth mentioning the analysis part because experiments are kind of useless without analysis. For our analysis, step 5, we're going to measure and compare the distance traveled by the solvent versus the distance traveled by the samples as compared to where our samples and solvent started. And the comparison of those distances is going to allow us to determine the different polarities within the different separated components of our mixture. You can also clarify all of this writing with a labeled diagram. It can never hurt your case, certainly always encouraged, and oftentimes AP readers will actually ask for diagrams. So drawing a quick little diagram like this in alongside your answer, along with your answer, will help clarify your points and could help eke out a couple points here and there. So never a bad idea to include a labeled diagram. So the last part of this We Do practice wants us to draw a chromatogram that would represent what we know about the 2 amino acids. So the first thing I'm going to do is draw my little chromatogram. So I'm going to draw my little square here, and then I've got my start line at the bottom, and then I'm going to do a little dashed line at the top to indicate where my solvent was allowed to run to, so kind of my end line at the top. And then I'm just going to do blue for my serine. So I'm going to draw a little blue dot down here at the bottom to represent where my serine started. And then I'm going to use yellow for my glycine. I'm going to draw a little yellow dot here for where my glycine starts. So now to know where these two are going to end up, I have to look at the polarity of the substances and how those polarities relate to both the mobile and stationary phase. So remember, our stationary phase, our silica gel or our alumina, are very polar. So that means our serine is going to have a higher affinity or more interaction with the stationary phase, with our gel, with our plate. And our solvent is typically going to be a nonpolar solvent. So that means our glycine, which is nonpolar, is going to have a higher affinity for our solvent and less interaction with the stationary phase, but it's going to have more interaction with the mobile phase. So don't get too hung up on trying to determine polarity based on the structures that we have here. These go much farther into organic chemistry and functional groups than we do, so don't really kind of stress out about these. You can actually effectively ignore them and just go with what they told us that the polarity is for these 2 amino acids in the question. So just take their polarity at face value. Don't stress the structure too much. So going back to our chromatogram, so looking at serine first, since serine is polar, and as we said, it's going to have a higher affinity for the stationary phase, it's not going to travel as far. So it's just going to travel a slight distance from the starting line, so we can draw it somewhere right about here. And then since our glycine is nonpolar, it's going to have a higher affinity for the mobile phase and less of an affinity for the stationary phase, so it's going to travel farther than the serine. So we can draw our little glycine ending point somewhere around here. And there you have it. There is a reasonable chromatogram that we could expect given these 2 amino acids, running them through a thin layer chromatography experiment.